I am so excited to talk to y'all today. As Courtney kind of indicated, I'm a huge fan of the Visible Body product. And part of that is because as an educator, frankly, some of what I was doing was getting a little stale. And this really helped me kind of find myself and try new things. And it's been just an absolute joy. So we're going to segue into teaching cell respiration and photosynthesis using VB Suite. If you're like me, this is kind of how you feel teaching cell respiration and photosynthesis. This is definitely something for me that always causes a lot of anxiety when I have to teach it. And in fact, and I'm going to just confess this because it's just, all of us here on Zoom, I even have notes to help me, my little cheat sheet, because that's how this works, right? Even as instructors, it is hard for us to engage all this information at the same time. But I think, and we'll get into this in a minute, that part of the problem is that we need to just refocus what we're looking at content-wise and how we're engaging with the content. And we do that, then we're going to make it more fun for us, and the students are actually going to remember it. And it's going to be a better experience for everybody. So when we go to the next gen science standards and we look at them, one thing that you'll notice is that they really kind of emphasize this idea of linking the photosynthesis and cell respiration. So like, what's this whole carbon cycle happening? And basically how are plants creating the things that we need, and we're creating the things that plants need in this beautiful symbiotic relationship. All right, so we're going to go through that, but what I want to do before we move forward is just take a second and talk about what do we want the students to learn. So what are those take-home messages we want them to have when we finish teaching about photosynthesis and cell respiration? And so, Courtney, I'm opening up the floor, and I lost my chat bubble, so I gotta find
That's
it.
okay. Everybody, if you have something to say, what do you personally want your students to learn when it comes to cell respiration and photosynthesis? You can pop those in the chat, and I will read them out, and Cindy will write them on the screen you see right now. We'll give everybody a few seconds. All right. Let's see. I want my students to take some, they're coming in fast, some transferable knowledge with them to benefit their future careers.
Good one.
To understand that the two need each other to occur. Let's see, it is complex.
Yes.
That photosynthesis is a conversion of energy. Yes. It's not magically turning sunlight into food, Amanda. I like that. Key stages and events, flow of energy, easy way to learn the stages. Inputs and outputs.
Mm-hmm.
Let's see.
All right.
I think that's
so
many. I'm so glad that everybody has, have something to say.
All right. So we've got a really good list going here. And if everything works, we're going to return to this list because I want to keep us accountable that we learn that we're making sure that we're going over all the things the students that we want them to learn as educators. And that's, to me, the most sort of that central issue. Okay. So moving from there, we're going to get back to our sort of enchilada that's going on. Now I'm hungry. And where I'm going to start is actually here with photosynthesis. The reason I'm starting with photosynthesis is that from the perspective of the app, it's actually a much more, it's much more complete. But I did notice as I was creating this, I originally was just going to go in the order that's in OpenStax, because sometimes that's how I roll. And it didn't work quite as well in that order. So I can kind of give you some ideas of how to make it work better this way. Although if you're like me, sometimes the students are like, why did we do chapter 11 before we did 3? And it's like, it works that way better. So I'm going to start with this first model, which is an overview of a leaf. So now we're zoomed out way far. We're going to look at what's happening in this leaf, and we're going to kind of talk about how this, there we go, what happens here. So these are just literally videos, me recording what's going on in the app. You're seeing your light hitting the leaf, the CO2 going in. In just a second here, we'll see the water getting sucked up. And if you zoom in on those water molecules, they do look like Mickey Mouse, just like they should. And then we've got where that leaf is growing. If you zoom in there, you'd see it's a sugar molecule. So this is a really cool thing. And what you can do in class is start very basically, have the students tell you what's going on here. And you can see I'm kind of zooming into different sections. And the nice thing about this particular app is you can start, stop, rewind. In fact, you just saw me rewind to get back to where the water molecules were so we could show them to you, so that you can make it work for a way to sort of stimulate discussion that's highly visual. And I think it meshes with how our students are today. They tend to be very visual. Okay, so you can have them explain what's happening in the video. I'm a huge fan of a card sorting task. So you'd give them a stack of just 3x5 cards. You can even print one of those PowerPoints with like 6 slides and cut it out to make it easy on yourself. And you can laminate them if you want to keep them year to year. And have the students put the cards in order. The ones that I would use are sort of the CO2, O2, H2O, glucose, and sunlight. And then I would print a couple that say in and a couple that say out, and just kind of have them go through those steps. And now all of a sudden they're knowing what's going in, what's going out. We can even go further and have like, what are the numbers? How many CO2, how many O2, how many glucose? And have that piece of the conversation come in and have them self-derive the equation. So inevitably, if you're like me, you somewhere in your bio course teach cell anatomy. And usually what I do is I'll teach like animal cell, plant cell, bacterial cell, compare and contrast moment there. But this is a great chance to kind of go back to that. I tend to teach energetics at the end, probably because I'm trying to avoid it when I'm writing the course, but in actuality, it's kind of where I feel like it fits. So here you can actually see a video of me dissecting the plant cell and specifically dissecting the chloroplast. And you can see all those structures that are inside the chloroplast. And you can have the students even, like, they can take a snapshot of this and they can label it. And that teaches, we discussed sort of teaching them transferable skills. Digital, like that ability to sort of manipulate an image and be able to make a presentation from it. That's one of those translatable skills we don't always talk about. You can also have them compare it with the animal cell. You can even do one of my favorite active learning activities is sort of loosely based on America's Next Top Model. You could have America's Next Top Cell and have them debate which of the cells is better at creating energy. And they're allowed to throw shade at each other. In that process. So, you only create glucose. They're allowed to do that. It's fun. They have a good time with it. Okay, so moving in, we started with the plant, we went to the plant cell. Now we're going into that chloroplast. We're still zoomed out pretty far. And again, in this model, we're going to see the light hit. We're going to see the water go in, oxygen come out. And now we've got carbon dioxide coming in and glucose coming out. And you'll notice this is pretty like distilled down process, but it's giving them that overview of what are the inputs and outputs. Then we'll get to how we get there, right? So when I look at this one and I think about how I would use it, one thing that I would ask is just what's happening here? What's going where? And what are the, okay, that thing, it's the two red guys that are stuck together. What is that? And just kind of ask them those questions just to get them engaged so that they're not just listening to you talk. And it's always funny. I feel like every time I do an active learning workshop or an active learning seminar, I always end up talking way more than I ever would in a classroom, which is kind of funny. I feel like you shouldn't talk about active learning, but it's what we do. All right. So you can ask what molecules are involved. You can have them, again, write the equation. What do you think the equation is? What does it look like? Check with your neighbor. Does your neighbor agree with you? Watch it again. And you can have them kind of work on that piece of it. Okay, so now we can go even more detail, right? So this is yet another model that all these are within the app, and it's just me taking screenshots from my phone. If you have these on computers in your classroom, your students could do the zooming and all of that. And by the way, in practice, they do it better than you do. They're amazing with these things. So now we've got the light coming in. And the cool thing about this model is now we're starting to see that photosynthetic pathway. We're seeing the light-dependent pathway specifically right now, and getting to see sort of that electron transport chain that's occurring. So this gets into a lot more detail than we had before. And that's what I like. I like kind of having those different levels of detail so we can keep distilling it down, right? We're all on the same page. We know that we're getting that carbon dioxide and water in. We know we're getting oxygen and sugar out. And now we're starting to ask, how do we do that? So you can create, and these are just some thoughts that I had. I don't know if folks are familiar with a jigsaw, but in a jigsaw, what I would do is I would have team light reaction and team dark reaction. And I would have these students each study what happens, give them some very simple prompts, and then have them teach each other and kind of go back and forth that way. And they tend to learn better. And I'm probably speaking to the choir here, or preaching to the choir here. They tend to learn better from each other than they ever do from us as educators. So just having that sort of peer interaction. And the great thing about it too is they can be like, I don't know what's going on here. And they can have those interactions as well. And even if you've got a student that is really struggling, they don't know what's going on, they're going to ask another student, they're going to get the answer for that from them. And that other student that's really doing great in your class gets everything before you even say it. That student's actually strengthening their understanding because they're having to learn how to explain it. Another transferable skill. Okay. Again, I'm a big fan of ordering events. And so that's something that I would do is what's going in and where are the hydrogens going? You can make those cards, you can put anything on them. I literally do an evolution lecture and it's just different animals. And we kind of work out the story of evolution and what came first just using our cards. Okay. So now I'm zooming in just on that same model. So if you took it on your screen and just whoop, you would get here. The great thing about this is you're seeing the light hit those antenna proteins. You're seeing the electrons go down the electron transport chain, and you're seeing the hydrogens popping off. And my favorite is that ATPase, and looking at how that hydrogen gradient is being used to kind of tag phosphates on to those ATPs. So in this case, what you could do, if you wanted to get all fancy, is you could ask your students to label things. And you could even use the app, and you could be like, oh, well, this right here, this is an ATPase. These are the antenna proteins. Right? And you could have them sort of come up with, what are these things? What do they look like? What are the names? And where are the electrons going? One thing I really like about this model is it does show the electrons kind of going down the slide. And when I teach this, I often use a roller coaster and I tell my students, hey, as we're going down, we're gaining energy. And in a very similar way, these we're releasing energy rather, in the same way these electrons are as well. All right. So in this case, you could label the proteins. You could have folks indicate gradients. Hey, there's a lot of hydrogens on the inside. Where are they going to go? They're going to go high to low because no one likes to use the stairs. We all like to go down the slide, right? And you can have them sort of summarize what's happening and maybe even discuss the energetic molecules. Okay, what's this NAD? What's its energetic state? What's the difference between ADP and ATP? And have your students figure out where the energy is in those different places. Okay. Oh, there you go. When we get to the dark reaction, again, they can talk about what the molecules are used and what process is occurring. You can have them break the different parts of this video into fixation, reduction, regeneration. And you'll see here, you can see those rubiscos coming in, combining with that carbon dioxide, right? And you can, what I like about this is you can kind of see where things are changing. And it does indicate, and will very shortly, that this is going to take 2 turns, right? There we go. And so you can have your students chop the video and say, oh, this is this part, this is this part. And they actually have to think about what is happening during that part to do that, even though the output's very different than what you'd think on like an exam question, right? I'm also added in here that one of my favorite resources to use when I'm teaching this piece is Photosynthesis Interactive through a source called Bioman Bio. This is just a website, and you're going to get it in your instructor kit. And the great thing about it is you're literally dragging the molecules. And so it kind of, again, gives that sense of what's being combined, what are the important parts of this. And it does, full disclosure, go in a little more detail than this. So it kind of depends how much detail you want your students to have. But I think if you have them start here and then you're like, okay, let's go one step deeper, then you could go to the Bioman Bio. All right. So we might think about labs, right? What are we going to do to have the students have a hands-on experience? And this was one of these like kind of crazy experiences where I was thinking, okay, the lab I do is this spinach leaf thing. And then I open the visible body handout, which I did not write, and it's the same lab. So basically you take a hole puncher to some spinach leaves, although you can use a lot of different leaves. Take a hole punch of your spinach leaves, put a little water with a little bicarbonate in it, knock all the air out of those leaves, and then you put them in a beaker under bright light. And the cool thing that happens is as they undergo photosynthesis, bubbles will come out, and this happens in about 5 minutes, and those leaves start to float. And so you can have your students literally just check in at different, every minute, how many leaf discs are floating. And they love this. It's very hypnotic. It's like watching a lava lamp. I just dated myself. It's fine. It's very hypnotic. They kind of enjoy doing this, but we can take it a step further. So what you have in the visible body handout is this basic lab, and then also like some different pieces, label parts of the leaf. These documents, last I checked, at least were Word documents, so they are things that you can kind of pick and choose what you want, and then get rid of the pieces that maybe you don't want or maybe won't fit in your class period. I added into this lab, and what I do is I use theater gels. And if you ever were in theater, theater gels are what they put over the lights to make them change color. And the way that they do that is they actually block specific pieces of the light spectrum. So you can literally get a graph of what parts of the spectrum are blocked by this light, because that's how theater people do it. And then you can have your students hypothesize which light is more important. Is the blue part of the spectrum more important to plants? Is it red? Is it green? And you can have them test this with those theater gels and look at how did the theater gel impact photosynthesis? Or really, how did the light spectrum do it? So what I do with this lab in my course, it's part of our assessment paradigm for graphing. And as a result, I've got a very simple sort of like, here's your checklist for everything that should be on your graph. And to me here, their output is their graph, and then they have to explain their experiment with their graph. And that's how I do the specific ones. They basically have to say, hey, this, red is better or whatever. Okay, it's not. But they have to explain which spectrum of light worked the best. And it gets them using that graphical literacy, which I think is one of the most important skills we give them in a science course. Okay, so moving on, we're going to go over to the cell respiration part. And I saw in the comments earlier, we were talking about what should the students learn, right? Respiration, cell respiration is linked to respiration. This is one of my things. I'm a physiologist at heart. That is my bread and butter course, as it were. And I want the students to know, well, this is why you breathe. We breathe in oxygen, we breathe out CO2. Where is that coming from? Where is it going? And then you can talk about mitochondria, and you can talk about, well, neurons have a lot of mitochondria. Why might they have that? Oh, because they've got a lot of those cell pumps. And you can talk about thermoregulation and having leaky mitochondria so that you use more ATP and you can make more heat, right? So there are a couple different aspects you can talk about in the physiology sphere if you set this piece up. And the amazing thing is the students will be like, well, respiration is used in both, and they won't make the connection. So I always like to make it deliberate. All right. So when we go to cell respiration, we can break it into several steps. This is my lazy instructor self coming out and breaking it into steps, just using a Wikipedia diagram. I found that I was like, hey, this is pretty good and it's pretty basic. And we're going to step through those. I'm not going to spend a ton of time. I just want to show you like different ways you can use this product, and then we'll open the floor up. So what I like to do with my students, I usually give them guided notes for my lecture between the like class session or video or whatever I'm doing and the book. They should be able to fill out these sheets, and then they just use them as their study guide. In one of those sheets is this kind of chart. This chart breaks it down into those inputs and outputs. And I was so glad someone said that, because that to me is like one of the biggest take homes, right? What is going in, what is coming out of each step, and how is it used? And then again, if you're more on the physiology end of things, this whole idea of is oxygen used? That becomes really important, right? Because you're like, y'all need to know what part that's used in, and where it's not used, we could put something else in. So if you wanted, you could probably put fermentation in here as well and add that in as a different part of the cycle, or a different potential piece. So when we look at glycolysis, what I have here is I've got your simple sort of diagram from OpenStax. I used OpenStax because the visible body product is aligned with OpenStax. And I suspect a lot of you are kind of engaged in this discussion of textbook affordability. And of course, OpenStax is like the ultimate in affordability. So when we look at this, we can look at this diagram and say, okay, ATP are going in, we're breaking a glucose into two things, and we've got 4 ATP coming off. And the cool thing is when we look at this model, these ATPs are going in, all right? And now we've got the ATPs coming off of that. We've broken into 2 sugars, which created those ATPs, and we've got our charged up NADs coming off of that too, which is, right, really great. And I like this because it's a little more live than that diagram that your students may not know how to read. So your students could fill this out, and what they would have is the starting molecule is glucose. The ending molecule, in my case, I usually get really easy on them, and I'm like, just tell me it's a 3-carbon sugar. Energy used, they would know that there's the investment of those 2 ATP. Energy released, they know you're getting 4 and the NADs out. And I didn't see any oxygen, did you? Okay, great, we're gonna say no on that one. So when you go further into OpenStax, you see this. This gives me nightmares. Okay, this leads to me having to have my notes, right? And I really think this is how I learned cell respiration. I am not a biochemist. In fact, I'm actually a really horrific chemist. So this is a terrifying thing. I think for most of us, we should very much avoid that. And if we do teach it, rather than focusing on, it goes from fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate. I can't even say those things right. We can talk about, well, you know, if we add on this phosphate, we destabilize the molecule, we destabilize it, it's going to break apart. If we add on our acetyl group, it's going to make this so that now it is hydrophobic, it can cross the cell membrane. If we can revert back to what the function of these things is, it makes it easier for the student than remembering these names that are hard to memorize. And are, this frankly very complex process that I need to get off the screen because it's given me palpitations. All right, so you get in your oxidation of pyruvate. What I wanted to show you here is that you've got your text that's in the app. Anytime you see the little I, you can click on that and it's got explanation text. You could have the students work through that and say, okay, what piece of the model is showing you this? And have them show you what it is. And then you know that they're interacting with that product, they're thinking about it. They're kind of applying their knowledge in a way, right? And then you can use it as a supplement to this, which is the OpenStax oxidation of pyruvate, right? It's showing that you're getting rid of that carboxyl group, you're adding that coenzyme A, and now you've got acetyl-CoA. I always get a little frustrated when they don't talk about it getting across the cell membrane, but it's because we do a lot with cell membranes and physiology. There we go. Here's what it looks like. And you can see, there we go. We tagged something different on it, right? Those purple things are different, and we freed up some carbons. Cool. What we want to avoid at all costs is this. This is the real Krebs cycle, right? And I think it's for students and instructors alike. You learn it and forget it. And that's kind of the way it is. And by the way, same for the Kelvin cycle. We go through that one too. So how can we take something like this and make it into something the students are going to understand? When I look at this from an app standpoint, the nice thing is it's showing me that those acetyl-CoAs are going in sort of one at a time. We've got energy coming off, and it's not a whole lot of energy coming out. I often tease my students. I'm like, I did this cycle twice and I only got this t-shirt, right? But one of the ways we can talk to our students when they look at this graph, if we want to work that in, is just say, follow the carbons. How many carbons are there? Okay, we're starting with 3, we're adding, 3 more. Now we've got a 6 carbon. Now, every time we take a carbon off, we're going to add some energy to something every single time. And you can kind of, one of my colleagues calls it like a taxi cab. And they're like, every time the taxi cab stops and lets out a passenger, there's some energy released. You can do it that way. And I think even if you want to get more complex, starting the students where it's simple and tractable, and then adding complexity is the way to go. Because otherwise, once they're lost, they're not going to hang out anymore. So you can go ahead then and break it down simply and just say starting molecules and ending molecules. But if you want to, you can actually point to specific elements. Okay. Now, when we get to the electron transport chain, this is one of the places where you're going to look at this model and you're going to be like, that is not the level of detail that maybe I want my students. At least if you're, depending on the course you're in, you might be like, there's a way around it, and I'm going to show you that. And it's actually really cool. So first off, if you're just interested in inputs and outputs, you're totally going to get that, right? We know that our NADs and FADs are going in, and all of a sudden we saw, oh, this energy and water come off, right? So we can figure out that there is definitely some stuff going in, and we can figure out what it is if we just sit and break down the app. But we can take it a step further. And one of the things you can do, and this is why I would recommend here, starting with photosynthesis, is that we have seen the electron transport chain in photosynthesis. And oh my goodness, hey, when evolution gets a really good idea, she sticks with it. We can see the process going on in the mitochondrion as well, and they look similar. So this is like a great chance to link these together and ask the students, hey, how would you make something that looks like this, but for the mitochondrion? What do you think it would look like? When would the different things come in and out? And now you've given them a project to do. And it's a project to do that's very sort of resource. They're able to kind of figure out what the different pieces are. And it's going to be good for them to do that sort of high level thinking. All right. Oh, I've got our video of our electron transport chain. I hit this one earlier, but I'll be honest, I can't get past the ATPase. It's just adorable. I don't know why I think it's adorable. It just is. One of the neat things that you can always talk about with the students as that because we're not recharging at photosystem 1, well, what are we going to do with those fads that come in lower? Oh, they just give off less energy. And so that's a good way to have those conversations. Again, you can relate it to the mitochondrion model. And here we're going to just quick mitochondria dissection, which you can do in the cell model. And you can kind of look at it. And if you're a physiologist like me, you're going to talk about surface area of that inner membrane and why that is so important, because we love membranes and all things crossing them. So I wanted to give you some ideas of labs because it really comes down to that hands-on, right? That's what makes biology fun. That's why it's everybody's favorite class. A lot of times when we are we're like, okay, I taught cell respiration, I need a lab. What's the lab we do? It's fermentation. And okay, there's a good part of that. Fermentation, beer and bread. Who doesn't love a good carb, right? So it's something that people love. They love to hear about. They're familiar with it, at least in some way, shape, or form. But the neat thing about it too is here it doesn't quite fit with what we were looking at, which is the aerobic process. So it's a supplement to it. You can kind of say, well, hey, here's a lab that's going to teach you what we can do if we don't have oxygen. And then you can look at fermentation. The typical one, you just put yeast and a couple different sugars in a test tube and you put a balloon over it and look at how much air comes off. But rather than that, what if we get a little more like to our lab, a little more use out of it? We can actually relate photosynthesis and cell respiration. We can say, hey, how are these things the same? How are they different? And we can have our students engaged in that process of what elements of these are similar. You can even do like the America's Next Top Model knockoff thing that I do, and you could be like, what's the most important protein? The truth is, it doesn't really matter. The important thing is that the student researches it and tries to figure out how it's different from everything else. And in the process, they learn about everything else. All right, so for this, there is also a lab through the visible body materials. It basically breaks down the app. They do a dissection of I believe, of a mitochondrion, and they kind of go through the process and it's like, what's coming out, what's going in? And you could have your students do this before class and then come in ready to discuss and do a flipped classroom, which is always fun. All right, I'm going to do a shameless plug. I have never been able to flip a classroom, but I do lean. And the way that I lean is I have the students watch videos beforehand. So in this case, what I would say is we've got a cell respiration video. I think it's in 2 minutes or less. And it just goes through the highlights of cell respiration. And you could have your students watch that and come to class prepared. Hopefully they like it. All right, I'm watching my time, Courtney. So I've got some other assignment ideas. One thing that I do in my class is have my students narrate videos. And so they can take the images from visible body and then they make a mini lesson. I limit it. And the great thing is that D2L is limited to a 5-minute video. So they're 5 minutes or less, which is really great. And then they have to tell me what the process is. And it allows them to get experience explaining scientific topics. You could also make an informational pamphlet, or what I'm going to show you is a comic. The difference between the two is the informational pamphlet is going to have more words. So I start with a rubric. You're going to get these, literally a checklist. I used to give my students a prompt. They never read it. So now we just give them this checklist. Hey, if you're explaining this, here are the pieces you need to explain. And here is actual student work. One of my students actually did this. I'm really proud of it. And what they do is they go through this process where they actually get critiques from other students. And we do that online through a program called VoiceThread, but you could do however you like. They can do it in person. It doesn't matter. But they get that feedback on, hey, I think to get full points, you need this. And here's one that scored sort of, I wanted to show one that was a little less good than the last one. So this one was not an A. But when you look at this one, you can see that the student really is kind of focused here on the carbons and what carbons going, how many carbons do we have and where? What are the different parts of the process? Even when it's not perfect, it's great. And even if your students are not great at drawing, they can still get a lot across. You can still do a lot with dots and circles. It's really more about them engaging with the material and thinking about what it would look like and not just regurgitating whatever they memorized. So if you wanted to take this further, you could do case study discussions. One example, there was a famous case in France. It's a little old now, it's from 2017, but it was Charlie Gard, who was a little boy who had a mitochondrial disorder. His mitochondria were dying. And so there was some question as to whether or not they should pull the plug. It's a tough discussion, but it's actually an interesting ethical dilemma about, why is that energy so important? And why is it, if this is hitting the nervous system specifically, that this child is non-viable? More fun stuff, you've got the leaf sheep. Leaf sheep are a nudibranch that has a symbiotic photosynthetic bacterium, and those things all live in the same animal. I'm a Midwesterner at heart. I love rhubarb. Rhubarb is another one. They grow it in the dark. They trick it into growing by warming it up. And the way that we get those beautiful long but thin stalks in the grocery store is because they grow that rhubarb in the dark and it's reaching up trying to find the light. So it's kind of a fun process to talk about with your students as to well, why does this work? Why is the plant trying to get to the light? How does it change the sugar balance in the rhubarb? And why would that make it more culinarily interesting to some? Okay, so coming into the finish, did we get it right? And what we're going to do is go back to our list here. And I want you to see that we did talk about a number of sort of transferable skills. To me, big ones are data interpretation, our discussion, like being able to discuss science and kind of break things down, thinking about what things look like and how you can make that explanation both verbally and visually. Plants and animals need each other. This is where when you do that photosynthesis and mitochondria right next to each other, it brings that home. Your leaf sheep example would be perfect there. I think we got that it's complex and we got the ins and outs. And I think I didn't cover it so much, but we could get into that photosynthesis is storing sugar and how that sugar is being stored. So I think we're hitting on the major things there, but I want to turn the floor over to you to ask questions and just to throw out ideas. I want to hear about the cool things that you do. So there we go. I'm going to stop sharing. Oh, stop sharing. There we go.
All right. We have a question, and remember, if you want to talk, just raise your hand, and I will unmute you. But we have a question from Anna. How well would this convert into an online course on the same topic?
So the great thing about this software is that your students have web access. So all of them have it at home. And I talk about this with anatomy instructors a lot because if you've got an anatomage table, it lives in a lab and they can't play with it. This is on their cell phone. It's like you've got them right where you want them, right? So what you could do, and this is what I do in A&P, I have the students record videos of them doing a dissection. They verbally walk me through. They practice it so many times to get it right that they know it really well by the end. And it works as an assignment. You can also have them, I've used Google Jamboard. I'll see if I can pull one of those up where they do the card sorting tasks online. And it's basically virtual sticky notes that they use to figure out what goes where. So there are all kinds of cool things you can do. You can even have them, if you were to use like the visible body labs, you could have them do that in the comfort of their own home. Hopefully that answered a lot of your question. So,
we
is there a
comment
visible?
about Bioman Bio games and a VB equivalent.
Sadly, not yet, but with immersive labs coming in, which is the new hotness, I suspect something along those lines would come in. They do go together really well, and you can link to those games very seamlessly if you're using courseware. And I've never had any blowback from students. They always love being able to do both, and they kind of like that that is a little low tech. They kind of dig it.
We do.
So the Chromebook workaround. The Chromebook workaround, what we do is that we actually have a virtual machine on campus. And what our students do is they're able to, with the Chromebook, log into the virtual machine, and then they're able to run Visible Body on the virtual machine. And that's sort of the Chromebook workaround, because we do have a lot of students that have those. And I don't think the app works, at least last I checked, on a Chromebook. So that's kind of how we do it.
It actually, I mean, it works on my Chromebooks. The thing about Chromebooks is that their configurations vary quite widely. But the best way, I think Kara had mentioned this, but it works on most Chromebooks. But the best way to see if it will work on yours is to just do a test, a quick test. And I actually, Anna, I'm going to drop a link in the chat to our support site where we talk more about Chromebook support. So you can see the specs, I guess the best configurations that we offer, and see if they jive with yours.
All right, and then I saw how to add the animations to PowerPoints. What I do is I actually take a video and I just add the video. The other way to do it, and this is like what changed my teaching more than anything, is that I started running, so I have my iPad, I always have my iPad right in front of me, and I started running basically the app on my iPad, and then I'll have QuickTime mirroring the iPad screen. And then I can rotate things around. I can speed it up, slow it down. I can make it look however that I want it to look for the students. We've even gone for a larger version of that at my university. Our tech guys had a huge touch screen that like no one ever used. And they said, Cindy, we know you can use this. And I have this, I think it's a 72-inch touch screen now in the lab, and we use that a lot too, because I can just show it to the students and very seamlessly use it like a giant tablet. It doesn't have to be a tablet. I just find for me, the tablet controls are a little easier than the web controls. Oh, Coco's here. Hi, Coco.
Do we have any other questions for Cindy, or any ideas that you might want to discuss a little bit further, or what you do in your class? Oh, we have a hand raise. Yay.
Yay.
Suzanne, okay, I am unmuting you right now. Suzanne, I think you might need to unmute yourself.
Gotcha.
There we go.
I'm not in the classroom, but I do K through 12 outreach, and via Lego, this is late 1990s, guys, I got a glucose modeling kit. Molly-Mod models came out. The glucose model kit for chemistry, for chemists, was a bust because of the rules of chemistry for high school students. However, we have amended it, and I did amend it with the teachers. And we do have the 6CO2, the 6H2O, making a glucose model, and you get the oxygen back. The fun thing would be with yours, because of the way you do the modeling in the class, the ultimate question to the kids is, where did the oxygen come from? And in doing the modeling, the way it works, they always answer CO2. So your visible body and your way of working through things would be a good kind of joining of that to kind of alleviate some of that, besides doing a full chemistry experiment or discuss the history of what happened in the 1800s when they devised where the CO2 was coming from. But I could see using parts of this aspect and strengthening for, say, an AP biology high school class, the parts of it as such. I do go through the more structural parts if you're doing it in chemistry for the students, because I use the models as, well, here's the model, but is the model biologically, chemistry good, or is there some good points and bad points to it as such? And you're using models in your stuff too, because all these are 3D models and moving models and everything, so it works out fine. But it's something that the kids find enjoyable, and they kind of work through it all. It does showcase some good points and bad points as far as modeling goes, but it's a learning tool.
Yeah.
And I use it as such. But I liked these visible bio. Well, I've used these guys when they were basically plastic kits when I was in 5th and 6th grade.
Yeah.
Showing my age.
Well, the great thing about this is that you don't have to like not use those kits or not use those ideas. In fact, I used to do a lab. It's your Lego lab. What I'm loving about it is you're going to be able to track the carbons. I used to do one, and it was literally just build a brain out of Play-Doh, which we use sometimes in physiology, but just what you can see it as like a supplement. So your student could have the app up, and they could be using the app to help them figure out which Legos should be present and what went away. And
Right.
it's causing them to kind of do that inquiry-based learning where they're like, do I get this? Do I understand what should be going on? And
Yeah,
then
it would be interesting to see you make the Lego kit your way, and literally what you end up doing is they build the 6-sided figure with the 1 oxygen, but in doing
yeah,
that, they've basically, they broke up all their carbon dioxides, and in physiology, that's not really what the cells do in that little chloroplast. So it'd be, if they could do that, they see that, then go back to yours and see what happens, and then maybe they could see with the Lego models trying to do it that way, follow it through and break it apart where the pieces go and put in. It might be an interesting kind of take on it for some students.
absolutely. Well, and I love to sometimes have them talk about, how can we make the model better? How can we make it explain this piece better? Because
Okay.
I think one of the problems we have is just what you're saying, right? This is a chemistry thing, and it looks like a chemistry problem. How do we make it reflect the biology and reflect it very authentically and clearly? And that's difficult. So I like this. And by the way, I took a second and just Googled it because I was like, otherwise I'll forget in 5 minutes. The absent-minded professor is real. And these kits are fantastic. And there's even an APS paper about it. That's exciting.
Well, I know the Molly-Mod does it. The only thing is the Molly-Mod models don't do with Legos are cheap. You can have the kids bring in their CO2s and H2Os, and you can get them started on it. You can play the game of how you make one and to make it more chemically relevant, et cetera, et cetera. So you have polarity. Oh, you can do plenty of things with the stuff. And I've got a five basic lesson thing on it already. But the idea of taking the models and following through some of the visible body parts of the different things would be, I think, really fun for the students to do, and they kind of bring home the points quite clearly.
Absolutely. And I like that you're getting like the visual and the tactile all at the same time, right? It's all the modes we can have them learn. Every part of their brain, right? Let's use it all.
Okay, thanks a lot.
Thank you. So I'm seeing about D2L or visible body linking in D2L, and I know you got a link about that. So it does, I will admit, so I work at Minnesota State, and the state, for whatever reason, decided that they didn't want to let us link it with D2L. So I actually use courseware, which is their own sort of learning management system. And I really like using courseware. It's not that hard for me to port grades from one to the other. And usually I just give my students the courseware link. And frankly, in some ways it's cleaner than D2L to be able to just set up a lesson and be like, boom, 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 boom. You're going to go from one to another. So that's an option as well. And I play around with it.
All right, we have 5 minutes left. Does VB integrate with Canvas? Yes, we do. I'm actually going to link to everything that we have for LMS integration in tomorrow's email. So you'll get all of the information about our various integrations then.
Very cool.
All right.
And I will say the way that this changes your teaching is just being able to like show the student a model and be able to talk. What do you see? What's happening? What do you think is going to happen next? How would we, and it changes you from death by PowerPoint to creating something much more conversational. And I'm very lucky in my classroom. All the students have, well, depending on the class I teach, a lot of them will have computers. And then they can look at it, at least in small groups, and start to talk about these processes and kind of dissect them. And Anna, I can tell you more too. So our anatomy and physiology used to be an in-person class with an in-person lab. And we started using visible body, and we found, and you can find the video of me talking about this, the grade gains were incredible. We actually call it the visible body effects. Our students, we had a 19% increase in A's, and that wasn't just my institution. I studied it across 5. I really need to get that paper written. But here's the thing. We, over the pandemic, switched it to an online course because we had to, right? When we switched it to an online course, all of a sudden, the students were still doing the labs. They were still engaged. They were still performing just as well. And we've done online synchronous and online asynchronous with our students are loving it and they're doing really well. So it definitely can be used. I think it's sort of an exception to sometimes what we see when we get publisher materials that are online. I won't mention certain companies, but sometimes we see materials that just aren't very good. This is something that the students, they get it, it's intuitive, and it's aimed really, I think the majority of it, your C students are going to get it. And your A students will take the supplement you give them, rather than a lot of the programs try to give so much that the A students are overwhelmed and your C students are like, rowing their boat out to the island so that they don't have to go to class because they're gone mentally from the materials. So these are fantastic. I've had a great experience with it in my class. And again, I looked at 5 different institutions, and we all had the same things to say. Oh, so did you. Excellent. Go, Anne. So I find that they do the assigned work, but I've changed how I do my deliverables. And I do have periodic, did you read it quizzes and that kind of stuff. But we also will have them turn in things. So I've been doing the comics and sort of these narrated videos, and they have to do those because it's their exam replacement. But they also have a lot of fun with it.
And actually, I did want to jump in and mention that Cindy, you actually did a webinar last year, the year before. I don't remember how time works anymore, about moving beyond exams and your kind of standard assessments. So I can link to that in tomorrow's email as well. It's definitely worth the watch. It's great.
Thank you. Well, and I'll give you my spoiler. I've been thinking about this a lot. Part of why I've been moving away from exams is your modern students are different than we were. We grew up with a card catalog. We grew up having to find information. Now it's everywhere, and they have to learn to edit. And so if you see the comics and you look at it and you're like, that's kind of odd, it's me trying to teach them note-taking, editing, and how do you order your ideas into something logical. And it's kind of giving them that process that I think we got very naturally through having to search for things in the card catalog and find a book and then figure out what the next book was, right? So I think that it's a way to build that sort of soft skill infrastructure into the deliverable that students naturally going to give. And at my school, we look at bias in grade data, and I'm happy to report that I don't see it in my data now that I've switched to these, because our students are just not as stressed about it. It allows them to kind of take time and breathe and think about what things should look like.
Okay, so it is 2 o'clock on the dot, which means that we are going to be wrapping up. So if anybody has any final questions that they want to sneak in, now's the time. But I just wanted to thank Dr. Harley again for joining us for this amazing presentation. And to remind everybody to keep an eye out for the email that you'll be getting tomorrow with all of the follow-up goodies, the recording of this webinar, which you will also be able to watch on our YouTube channel. And I just wanted to thank you all again for joining us today. Okay. So we will see you next time for our next webinar sometime later on this spring. And Cindy, thank you so much again. It was wonderful as always. Alrighty. Bye, everybody.